My name is Gail Clark, and I'm here with Ag in the Classroom to show you how you can bring the world of DNA to your students. Even middle school students with just a few simple materials can extract DNA from plant material. I know that sounds really complicated, but it is not. Most of you know that in most cells that there are 2 copies of each chromosome. Well, in a lot of plants you have a situation called polyploidy where they have more than 2 strands of DNA. That's the case in strawberries. So we have a giant amount of DNA to be extracted. And what we're going to do today then is we're going to take just a little Ziploc bag and take some freshly sliced strawberries. To this we're going to add some salt, and then we're going to begin a squishing process. Just close it up and start squishing. What we're doing here is not only physically breaking down the strawberries, but the salt plays a role. The sodium from the salt is going to stop an enzyme, or going to denature an enzyme whose job it is to break down the strands of DNA. All right, once we get those mushed just a little bit, we're going to add some water to make this mixture just a little more liquidy. Once again, close it up and let the kids squash. All right, now we're going to transfer some of this mixture to any kind of vial or test tube. You want to try to get mostly the liquid parts instead of the big chunks of strawberry. So at this point in time, if you would like to run it through some type of a filter, you may. Just going to use a pipette, and we're going to fill your container about 1/3 of the way with the strawberry mixture. Okay, your Ziploc bag can now be closed and just put to the side. All right, the next ingredient that we're going to add is a little bit of dish detergent. Couple of things about this. It only needs a couple of drops, and you really do want to tell your students that they don't want to mix it up so that it gets foamy. What we're doing here is just mixing it very, very gently. I don't want a lot of soapiness. What's happening here is the dishwashing detergent is breaking down the nuclear membrane where the DNA is stored. The DNA nuclear membrane is a phospholipid bilayer, and detergents are going to break that down. Okay, the next thing we're going to add is a very, very, very little bit of meat tenderizer. Meat tenderizer is an enzyme, and in meat, it breaks down the cell membranes, and that's exactly what it's going to do here and allow us to release the DNA. Once again, we don't want to shake this up, just stir it very, very easily. Okay, at this point in time, you're probably going to want to let this rest for a few minutes and let the detergent, the enzymes, and the salt do its work. Well, we're back. We've waited about 5 minutes, and we have one more thing to add to our mixture before the DNA becomes apparent. To our mixture right now, we're going to add rubbing alcohol. I've kept it in this beaker of ice because I wanted to keep it cold. When we add the alcohol, please make sure that you keep your test tube slanted. We want to slowly pour the alcohol on it and put about the same amount of alcohol in that you have in your mixture. We don't want the 2 of them to mix. Instead, what we're interested in is the interface between the alcohol and the strawberry mixture. The alcohol is going to actually dissolve the water away from the DNA strands. If we keep this slanted, the surface area between the interface has a larger surface area between the alcohol and the strawberry mixture. What you're going to see, and this could take a few minutes, but you're going to start to see strands of DNA. It will be white. You can take a splint like we're using here today, and we're going to gently bring some of the DNA up into the alcohol. Where more of the water will be dissolved away from it. Okay, once again, now this is going to take a few minutes. The students can just rest the test tube in a small jar like I'm doing here, or they can even put it vertically in a test tube rack. Okay, we've waited just a few minutes. If you take a look up in the alcohol part, there's some white blobs, which is the DNA. There's also some DNA down in the mixture. We're going to take it out and try to put it on here and show you a little bit. This is called spooling. Let's see what else we can get out of here. Let me see. Bottom line is, we have extracted DNA from the strawberries. We've seen how you can use a variety of plant material to extract DNA. It's easy, it's fun, it's interactive. I invite you to download the lesson from Ag in the Classroom. It's a great experiment. Your kids will have DNA that they can put under the microscope, that they could keep in alcohol, and they can go home, and when mom says, "What'd you do in class today?" you can say, "I extracted DNA." Pretty cool.